Prader-Willi syndrome is an incredibly rare disorder, only affecting 1 in every 15,000 births. Its distinguishing symptom is hyperphagia, or extreme hunger coupled with the inability to feel full. While Prader-Willi syndrome is little known to most, looking into the way it occurs can teach us a lot about the processes involved in many genetic disorders. The genetic alterations that occur in order to cause Prader-Willi syndrome manifest in the unstable 15q11q13 region of chromosome 15, also called the Prader-Willi syndrome critical region, or the PWCR. However, what makes this disorder so unique is that it can occur in any one of 3 ways, all showcasing important processes when it comes to genetic disorder inheritance. The first is deletion. This is the cause of about 70% of all PWS cases and is the deletion of the PWCR region, the paternal chromosome 15. During meiosis, the process that creates gametes, or sex cells, homologous chromosomes are aligned next to one another so that genetic material can cross over. This is typically an advantage, as the exchange of genetic material creates genetic diversity. However, all this shuffling around is also prime time for mutations to occur. Mutations result in the largest number of genetic disorders altogether. The second way Prader-Willi syndrome can form is uniparental disomy, which is responsible for about 30% of all PWS cases. This is when the offspring receives 2 chromosomes from one parent, in this case 2 chromosome 15s from the mother. In most cases, uniparental disomy in a chromosome has no effect, as the maternal and paternal chromosomes should be mostly identical with mostly identical functioning. However, uniparental disomy is a common cause of several other genetic disorders. Why? We'll come back to that in a second. The third and final way Prader-Willi syndrome forms occurs in an estimated 1 to 3% of all Prader-Willi cases and is caused by an imprinting center defect. So let's get into genetic imprinting for a second. Genetic imprinting silences gene expression not through deletion or other mutation, but through a process called methylation. This could be a whole separate video, but for now, all you need to know is that methylation puts a chemical tag on a sequence of DNA, which causes it to become inactive. Normally, the mother's PWCR is imprinted and the father's is expressed, but due to a tiny deletion in the father's imprinting center on chromosome 15, the father's PWCR is also methylated, so neither of the PWCRs are active. Taking a step back, this is also why maternal UPD has such an effect on chromosome 15. Not all chromosomes are imprinted, so there will be no effect if UPD occurred on a non-imprinted chromosome. But because having 2 maternal 15s means both PWCRs present are inactive, there's going to be some serious effects relating to the gene that section would code for, and that serious effect is Prader-Willi syndrome. Deletion, UPD, or imprinting defect, the common thread is the father's PWCR becomes inactive in some way, either through being removed, methylated, or not even there in the first place. So despite the 3 different overall processes, the core of the disorder stems from that one inactive section. So there you have it. Examining Prader-Willi syndrome teaches us about the crucial processes present in nearly every genetic disorder: deletion, uniparental disomy, and genetic imprinting. I encourage you to do your own research because, as we've seen, there is so much to learn from even the rarest of disorders.